Who is it?
It's the plumber.
Plumber. I didn't call a plumber.
Colorectal cancer is one of the most common cancers worldwide and a leading cause of cancer-related deaths. To understand how it develops, we need to look beyond the clinical features and focus on the molecular pathophysiology that drives tumor formation. First off, it's essential to distinguish between oncogenes and tumor suppressor genes, two types of genes that control cell growth in opposite ways. Oncogenes are like the gas pedal of a car. Normally, their precursor forms, called proto-oncogenes, help cells grow and divide in a controlled way. When a proto-oncogene is mutated or overactivated, it becomes an oncogene and drives uncontrolled cell proliferation, contributing to cancer. Examples are KRAS, BRAF, and MYC. Tumor suppressor genes, on the other hand, are like the brakes. They normally prevent cells from dividing too quickly, repair DNA damage, or trigger apoptosis if the damage is irreparable. If these genes are inactivated or lost, the brake fails and cells can grow uncontrollably, also leading to cancer. Examples are the APC gene, the p53 gene or guardian of the genome, and the retinoblastoma gene. In short, oncogenes are mutated accelerators that push cell growth forward. Tumor suppressor genes are brakes that normally restrain growth but, when lost, allow uncontrolled proliferation. Cancer typically arises from a combination of accelerators stuck on and brakes failing. So, colorectal cancer doesn't appear overnight. Instead, it arises gradually through the accumulation of genetic and epigenetic changes that transform normal colon tissue into cancer. But what actually causes these mutations? Let's break it down. First, we have inherited predispositions. In conditions like familial adenomatous polyposis, patients are born with a defective APC gene. This is the so-called first hit. Similarly, in Lynch syndrome, patients inherit defective mismatch repair genes, leading to microsatellite instability. These inherited mutations set the stage for cancer development. Second, we have somatic mutations. These are acquired during a person's lifetime. Normal colon cells divide frequently, and with each division, there is a small chance of replication error. Over years, these errors can accumulate and affect key genes like APC, KRAS, and p53. Third, environmental and lifestyle factors play a major role. A diet high in red or processed meat and low in fiber exposes the colon to carcinogens like nitrosamines and bile acids. Alcohol and smoking increase DNA damage through reactive oxygen species. And chronic inflammation, such as in ulcerative colitis, further increases mutation rates by driving constant cell turnover. Fourth, we must not forget epigenetic changes. In the context of colon cancer, epigenetic changes refer to heritable alterations in gene expression that occur without changing the actual DNA sequence. Instead of mutations, these changes involve mechanisms like DNA methylation, histone modifications that alter chromatin accessibility, and abnormal expression of microRNAs that regulate gene translation. Together, these epigenetic events can switch off tumor suppressor genes or activate oncogenic pathways, contributing to colorectal tumor development alongside true genetic mutations. So now we understand what brings about mutations in the colon, a mix of inherited predispositions, acquired DNA errors, environmental exposures, and epigenetic changes. But how do these mutations actually drive the step-by-step transformation from a normal colon cell into cancer? This brings us to the adenoma carcinoma sequence, the classic model that explains the genetic hits, one by one, that turn healthy tissue into invasive carcinoma. Colorectal cancer often develops through a stepwise process called the adenoma-carcinoma sequence, also known as the Vogelstein model. Let's walk through how normal colon cells gradually transform into invasive cancer. The process begins with loss of the APC tumor suppressor gene. APC normally regulates the WNT signaling pathway and keeps cell growth under control. When APC is lost, cells start proliferating excessively, forming an early adenoma. Next, mutations in the KRAS oncogene occur. KRAS drives uncontrolled growth and survival of these abnormal cells, pushing the adenoma to grow larger and more dysplastic. Later, tumor suppressor genes such as DCC and SMAD4 are inactivated. These normally act as brakes through the TGF beta pathway. Their loss allows the adenoma to progress into a more advanced late adenoma. The final step is loss of p53, the guardian of the genome. Without p53, damaged cells are no longer repaired or eliminated, and the adenoma transforms into an invasive carcinoma. So far, we've looked at the classic adenoma-carcinoma sequence, the stepwise accumulation of APC, KRAS, and p53 mutations that drive most colorectal cancers. But this is not the whole story. Not all colon cancers follow that linear path. In fact, up to a third of cases arise through a very different mechanism, the serrated pathway. The serrated pathway begins with sessile serrated adenomas or polyps, which have a distinct sawtooth or serrated architecture under the microscope. Instead of chromosomal instability, like in the adenoma-carcinoma sequence, the serrated pathway is driven by epigenetic instability, mainly through widespread DNA methylation. This is called the CpG island methylator phenotype, or CIMP. The hallmark mutation here is BRAF activation, which promotes abnormal cell growth. In addition, hypermethylation of the MLH1 promoter silences DNA mismatch repair genes, leading to microsatellite instability. Together, these changes allow serrated polyps to bypass the traditional stepwise process and progress more rapidly into carcinoma. Clinically, cancers arise from the serrated pathway are often found in the right side of the colon, and they may present at a more advanced stage because sessile serrated lesions are harder to detect during colonoscopy. So, in contrast to the left-sided APC-driven adenoma-carcinoma pathway, the serrated pathway is typically right-sided, BRAF mutated, and epigenetically driven. So far, we've covered two main routes, the classic adenoma-carcinoma sequence and the serrated pathway. Both involve stepwise genetic or epigenetic changes, but there's another important mechanism to understand: microsatellite instability or MSI, which is often seen in Lynch syndrome and some sporadic colon cancers. Microsatellites are short, repeated sequences of DNA that are prone to errors during replication. Normally, mismatch repair genes or MMR genes like MLH1, MSH2, MSH6, and PMS2 correct these errors. When these MMR genes are mutated or silenced, replication errors accumulate unchecked. This is what we call microsatellite instability or MSI. Cells with MSI can rapidly acquire mutations in genes that control growth and apoptosis, driving cancer development. In Lynch syndrome, patients inherit a germline mutation in one of the MMR genes. The second hit occurs somatically, leading to high-level MSI and early-onset colorectal cancer, often right-sided. Lynch-associated tumors typically do not go through the traditional adenoma-carcinoma sequence. Instead, they can progress rapidly from mucosal to carcinoma due to the high mutation rate caused by defective mismatch repair. Sporadic MSI tumors are usually caused by epigenetic silencing of MLH1, often in the context of the serrated pathway. So, while Lynch syndrome is hereditary, sporadic MSI is usually acquired, but the result is the same: defective DNA repair, accumulation of mutations, and tumorigenesis. As we wrap up, it's helpful to highlight two major signaling pathways often involved in colon cancer: WNT and TGF beta. The WNT pathway acts like a growth switch for cells, normally ensuring that cells divide only when needed. In many colon cancers, mutations in APC or other WNT components leave this switch stuck on, causing excessive uncontrolled cell proliferation. Think of it as a faucet that won't turn off. In contrast, the TGF beta pathway normally functions as a safety monitor, telling cells to slow down growth or self-destruct when something goes wrong. When this pathway is defective, cells ignore these stop signals and continue dividing, similar to a broken traffic light that allows everything to keep moving. Both the WNT and TGF beta pathways illustrate how imbalances in growth control can drive colorectal cancer. You can think of WNT as a stuck gas pedal that keeps pushing cells to divide, while TGF beta is like a broken brake signal that fails to tell cells to develop or self-destruct. Together, these signaling problems, along with genetic mutations and epigenetic changes, explain why normal colon cells gradually transform into cancer over time.
Bye! Hello?